Dear students and viewers, welcome to the Veterinary Entomology lecture series. This lecture has been prepared for the student of third year first semester studying Doctor of Veterinary Medicine. The course name is Veterinary Entomology. I am Dr. Alim, Associate Professor, Department of Pathology and Parasitology, Chattogram Veterinary and Animal Sciences University, Bangladesh. So in this video lecture, I'm gonna talking about the morphological features of the flea and morphological adaptation of the flea, which enables them to move through the body coat of the animals very easily. Later on, I'll talk about the behavior and ecology, different flea species that are important for the man and animals, life cycle, and obviously the harmful effect of flea infestation on animal as well as man. And another important thing that is vector importance of disease transmission by different species of flea. And finally, I would like to conclude my presentation by highlighting the diagnosis, that is identification of different flea species and control of flea. Phylum Arthropoda is divided into different classes. So one of them is Insecta. And these classes further divided into two subclasses. One of them is Pterygores and another one is Pterygores. So Pterygores are those arthropods which have wings on their body. On the other hand, Pterygores are those arthropods which don't have wings on their body. For an example, louse and flea. So in this video lecture, I'm gonna talk about the order Siphonophthora, that is flea. So what is flea? Flea is a very tiny external parasite measuring 1 to 5 millimeters in length, dark brown in color, wingless, and laterally compressed body. Remember, this is laterally compressed, not dorsoventrally flattened. And they are less permanently ectoparasite or temporary ectoparasite because they frequently leave the body of the host. And the body of the flea comprises three portions. The first one is head, and the neck, and then abdomen. On the head region, the first organ is eye. So the eye is not a compound eye. This is a very simple eye. Sometimes instead of the eye, there should be a spot which is called photosensitive spot. And next to eye, there is a very tiny antenna, three segmented, and it is located on the antennal fossa. And on the head region, there is another important structure which is called genal comb. So genal comb actually consists of different spines. Depending on the size and the number of spine, some flea species can be identified. And next, and mouth part, I'll talk about the mouth part later on. And after head, the neck is located. Neck is divided, or there are three segments on the neck region. The first one is pronotum, middle one is mesonotum, and the third one is metanotum. So on the pronotum segment, there is another structure which is called pronotal comb, and depending on the size and number of the genal and pronotal comb, some species of flea can be identified. And on the neck region, there are three pairs of legs, and the third pair is heavier compared to the first and second pair. As a result, the fly can jump from the host body or on the environment. And abdominal region, there are 10 segments on the abdominal region. So tergum 1 to tergum 10. One can easily identify 8 segments externally, and on the 9th segment there is a sensory structure which is called sensilium. So this is the function of sensilium is very interesting. It helps the flea to detect the air movement, vibration, and the temperature temperature of the host, temperature of the environment, and odor of the carbon dioxide and urine. It has some role in the it has some role during copulation, and that's all. So on the body of the flea, there are hair-like structure. Hair-like structure. They are known as setae. And depending on the size and depending on the shape of the body, one can easily find the sex of the female. So if you look at this picture, you can see the dorsal surface is curved and the ventral surface is also curved. And if you look at the other picture, that is this one, you can see the dorsal surface is almost flat, but the ventral surface is greatly curved. So for the first picture, if you can see the dorsal, both the dorsal and the ventral surface is curved, you can consider the sex of the flea is female. But if you get the ventral and the dorsal, the dorsal surface is almost flattened or flat, and the ventral surface is greatly curved, in that case, the sex of the flea would be male. And in this picture, this is the spermatheca part of female genital organ, and this one is the claspers part of male genital organs. Mouth part and the feeding mechanism. Mouth part includes a maxillary pulp and a labial pulp. There is no mandible. And labrum epipharynx, the grouped one, is very developed one, and there are two other lacini. So with the help of two maxillary lacini and the labrum epipharynx, stylet or fascicle will be formed. And there is a hypopharynx which is very short, and the mouth part is overall piercing and sucking mouth part. The function of the maxillary pulp and the labial pulp in the feeding mechanism is to locate the feeding site. And when the fascicle is pushed towards the feeding site for sucking the blood, the cutting action is done by the maxilla because they have the tooth-like structure. And afterwards, from the hypopharynx to labrum epipharynx, the saliva will come through the feeding side, and on the same way the blood will be sucked up. So there are two pumps work here. The first one is cribarrial pump, which will suck the blood after the pharyngeal region, and later on from the pharyngeal region to the midgut, blood will be pushed by the pharyngeal pump. And most of the flea feed only for a few minutes before moving to another part of the host. Morphological adaptation of a flea or morphological modification. So due to the morphological modification, flea gets some benefit over a host. So what are those morphological adaptation? The first one is the body is laterally flattened or compressed. As a result, the flea can move through the body coat of the animal very easily. And the second characteristic is third pairs of the legs are heavier compared to the first and second pair. As a result, the flea can jump. So they can jump from the host body to the environment or one place to another place. One of the important features has been observed in case of the Genopsila chiopis. So these flea can move even after 31 centimeter in a single jump. And the next morphological adaptation is short antenna. So we know that the short antenna is recessed or located on the antennal fossa. As they are not erected, so they don't create any friction during movement through the body coat of the animal. And the third and the next another feature is setae and the combs. So you know the setae, hair-like structure distributed all over the body of flea, and they are directed backward. And the genal comb and the pronatal comb, they are also directed backward. As a result, these structures don't create any friction during forward movement, and Very interestingly, these setae and combs impede the flea being dragged backward by the host activities. And if you look at this picture, you can see the body is carved, uh, that is dorsal, that is the dorsal side, and the ventral side is carved. As a result, they don't create any friction, or they helps in the forward movement of the flea through the body coat of the host. And some of the flea, they are also known as the burrowing flea. They can burrow the skin, and they will attach at the side of attach there for a considerable time by the aid of barbed mouth parts. So these are the morphological modification by which the flea can get some benefit over a host. Behavior and ecology of flea. Flea are non-host specific, that is, they infest a wide range of mammals and birds. They are less permanently ectoparasite, that is, they frequently leave their host. It has been observed that the Genopsila chiopis species can jump even up to 31 centimeter in a single jump, and on an average it is 24 centimeter. With the exception is the stick-tight flea. This stick-tight flea permanently attached to the host skin, that is, they will borrow the host skin and will stay over there. The adults are parasitic, both male and female are the bloodsucker, and larvae feed on the debris of the host or on the feces, that is, flea dirt of the adult flea. Female are oviparous, female lay eggs on the ground, that is, on the environment or on the host body coat. The life cycle is complete, that is, the different developmental stages of the life cycle of the flea is different to one another, and lifespan is around 1 to 2 years, and flea activity is higher in summer season compared to other. Important flea species includes Tenocephalus canis, Tenocephalus fellis, Pulex irritans, Genopsila chiopis, Echidnophaga gallinaceae, Tunga penetrans, Spinopsilus caniculi, another family Pulicidae. So Tenocephalus canis, this is also known as dog flea. This mostly infect dog, cat, and different other animals. Tenocephalus fellis, this is known as cat flea. This infests cat and dogs. Pulex irritans, the common name is human flea. They mostly infest man, dog, cat, and different other animals. Genopsila chiopis, this is very important flea species. They are also known as oriental rat flea or tropical rat flea due to their distribution in the oriental or tropical regions of the world. Echidnophaga gallinaceae, they are most commonly found on the comb and wattles of the birds along with different other animals, and they are considered as stick-tight flea or burrowing flea because they can burrow the skin of the host and stay over there. Tunga penetrans, this is very important for the man, uh, and the common name of this flea is the jigger flea or the human jigger flea or the sand flea. And another one, rabbit flea, this is Spilopsilus caniculi. They infest the ear of the rabbit along with different other animals. And under the Ceratophilidae family, there are a few important species. The first one is Ceratophilus gallini. This is one of the commonest poultry flea. Infest poultry and different other animals, including man. And Nosopsilus fasciatus, this is known as northern rat flea. This infests rodents, man, and different other animals. Life cycle of a flea. So you can remember there are three types of metamorphosis. One is simple metamorphosis, another one is complete or complex metamorphosis, and the last one is ametamorphosis. So complete or complex metamorphosis can be seen in the life cycle of a flea. So complete metamorphosis, that is the developmental stages, adult, larva, pupa, they are different to one another. There is no morphological similarity with the adult with the larva or larva with the pupa. And after the mating, the female flea will lay eggs. They will lay around 300 to 400 eggs in total, but at a time around 15 to 20 eggs. And within 1 to 2 weeks, these eggs will hatch to larva. And larva requires 2 molting to reach pupa. That is, in the first molting, they will become larval nester 1 to larval nester 2. And then in the second molting, they will reach to, that is, they will molt to larval nester 2 to larval nester 3. And they have a mandibular gland, and with the secretion of the mandibular gland, this will attract the surrounding debris or substrate, and it will form a protective layer which is known as cocoon. So within this cocoon, the that is pupa will remain around 2 to 3 weeks, and afterwards they will emerge after breaking the protective layer or cocoon. And to complete this life cycle, it will take around 30 to 70 days depending on the temperature and humidity, but in hot temperature it may take even after 2 years to complete the life cycle. Now the question is, what about the life cycle of stick-type flea, for an example Echinophaga gallinaceae or Tunga penetrans? We know that these flea burrow the skin of the host and they will remain there throughout their life. So during this stage, there will be some morphological changes, that is extensive morphological degeneration, and this sort of growth is called neosomy. Neosomy means neo means new and somy means body. So this their size is very smaller, and afterwards there will be morphological changes and they will suck enormous amount of the blood, and the body will be look like this, and this sort of growth is called neosomy, and it actually involves major integumental chitin synthesis during the adult stages, and the genital opening of the female flea protruded through the pore of the skin of the host to facilitate the mating with the free-living males. And after fertilization, after mating, the female will lay fertilized eggs and they will drop down to the ground. And on the ground, the egg will hatch to larval stage 1, larva, and it will require 2 or 3 molting to reach to the pupa, and afterwards pupa will reach to the adult stage. So there are different developmental stages in the life cycle of a flea. I would like to discuss a little bit about those developmental stages. So it is known to you that female, after mating, the female will lay eggs. Eggs are very small, 0.1 to 0.5 millimeter in diameter, and they are ovoid and white in color. And this egg will hatch to larva with the help of the hatching spine. And these larvae are legless, eyeless, possess a well-developed head capsule and twin mandibles. They also possess a mandibular silk gland. With the help of this mandibular silk gland, they will produce silk to produce cocoon. I will show the picture in a moment. And what about their feeding? Most of the larvae feed on the organic matter in the nest or bedding materials of their host, or the feces of the adult flea, that is also known as flea dirt. So this is the flea dirt, and this picture indicate that is the larva feed on the different organic material or the flea dirt. And there are 13 body segments with there are 13 body segments in the body of the larva, but there is no distinction between the thoracic and abdominal segments, and there is no appendages, that is there is no legs on their body. And in the developmental stages there are three larval instar. One is larval instar 1, 2 and the 3. So in the first molting there will be larval instar to larval instar 2, and in the second molting there will be larval instar 2 to larval instar 3. Larva will become pupa after 2 molting, and as mentioned earlier, larva will possess a mandibular seed gland. As a result, they can secrete silk or adhesive things to attract environmental things like environmental debris, environmental materials or substance to make a protective layer around the pupa. So this structure or this protective structure is called cocoon. This cocoon helps the pupa, protects the pupa in different way. The first one is pupa, they are very much susceptible to the desiccation. So the cocoon helps to get rid of the evaporation of moisture or water from the body, number one. Number two, they also helps to get rid of or avoid the predators, because this cocoon helps to camouflage uh, with the environment. And the head tubercle, pupa possesses a head tubercle. With the help of this head tubercle, they emerges from this protective layer or cocoon. And afterwards, this stage of this pupa is called true pupa, because uh, true pupa means you can see the appendages. For an example, the mouth part and the legs can be seen externally. So harmful effect of flea infestation in different pet animals. The first harmful effect or deleterious effect is the production of anemia in adults as well as young animals. So this is not, anemia is not very important, maybe not very important for adults animals because they can easily compensate. But this is very important for the young kittens and puppies. They may not compensate the blood loss through electrophoresis. And the second deleterious effect is called flea allergy dermatitis or FAD, F-A-D, FAD. So how this is produced? During feeding, flea bites the host and releases saliva into the host skin. And in this saliva, there are different antigens, but they are not complete antigens. They are considered as haften. So this haften later on binds with the host collagen to become the complete antigen. And this complete antigen will produce immediate type of hypersensitivity. But this is noted that you may not get immediate type of hypersensitivity or type 1 hypersensitivity at the first bite. So this can be seen in the sensitized animal. As a result, there will be intense pruritus and animal will become very restless. Due to the continuous discomfort, due to the itching, animal will try to get rid of this sort of discomfort or restlessness. So what they will do, they will go for scratching or grooming or biting or rubbing with the fixed objects. In the meantime, there will be secondary bacterial infection, which and combinedly or all together they will lead to severe dermatitis, pyoderma, and alopecia of the affected region. And there will be also skin thickening. And in case of the cat, there will be a small papule associated with pruritus, and this is also very discomfort situation. There will be intense pruritus as well as the cat will become restless. As a result, they will do the same thing, that is scratching, biting and rubbing, as well as there will be secondary bacterial infection. So in cat there will be seen miliary dermatitis. So dermatitis can be seen in the affected throughout the affected body part. And in this picture, this is, you can see there is pruritus on the snout of a dog due to tunga penetrans infection. Harmful effects of flea infestation in birds. So you can see Echidnophaga gallinaceae on a rooster. They usually infest the comb, wattles around the eye and around the beak. So in both of the picture, you can see a lot of flea on the affected region. And this Echidnophaga gallinaceae, this is very important for the poultry because these flea can burrow the skin and they will produce a nodular lesion followed by swelling and ulceration of the site of attachment. And they will also, that is this flea will also suck enormous amount of blood. As a result, there will be anemia. So due to the local nodule formation as well as blood sucking activity, the young birds can be killed. Flea infestation in human or public health significance. Flea bites cause intense irritation for several days. Bites are characterized by a tiny purplish spot, and there are two types of spots can be seen. The first one is purpura pellicosa. This is the only purplish spot without any swelling. And this one is roseola pellicosa. This is due to the flea bite and surrounded by a slightly swollen skin. And in this picture you can see the tungiasis. If it is a lesion related to the tungiasis, you can see a black spot in the middle. And in this picture you can see flea releases a huge number of eggs from the affected site. And in tungiasis, this sort of lesion can be seen, and this sort of lesion is more common in people walk barefoot. Public health significance of flea infestation. So they produce annoying bite. As a result, there will be dermatitis, allergic reaction, and lesion on the skin. And due to the biting, there will be some discomfort situation, or due to this lesion, the discomfort situation exists for several days. And in tungiasis or burrowing flea, there may be found painful lesion on the skin, feet, and toe. Flea can act as the intermediate host for the completion of life cycle of different tapeworm, and I'll talk about the vector importance in a moment. And flea has another very significant role in the transmission of different diseases. The first one is plague, and this disease is also known as urban plague, rural or sylvatic wild rodent plague, La Peste, Black Death, or bubonic plague. And there are different forms of the plague. You can uh, note it down as a side note. So the first one is bubonic plague. This is most common in human, characterized by enlargement of lymph node, particularly the axillary and the inguinal lymph nodes. The second form of the disease is called septicemic plague. In this case, pathogen moves to deeper tissues from lymph nodes, and organism in blood and capillary wall start to leak, turning the color of the skin black. As a result, or therefore they call it Black Death. The third form of the disease is called pneumonic or plague pneumonia. This is one of the most life-threatening forms of the disease. In this case, Yersinia pestis infect the lung, and the organism can be shedded through coughing and sneezing. So, without aggressive medical intervention, this form leads to the death of the patient, and patient die due to the exotoxin or endotoxin released by the dividing pathogens. And other role in the transmission of the diseases includes murine trypus and tungiasis. So as mentioned earlier, flea has a very significant role in the transmission of different disease producing agents. So they can transmit the virus, bacteria, protozoa, different nematodes, parasite, and cystores. So the virus, the first one, they transmit the myxoma virus and it causes myxomatosis. The host is rabbit and the distribution is Europe, and Australia. Another common disease is Q fever. The organism is Coxella barneti, and this organism is transmitted by different flea species, and the host is mammals. And the most important uh, organism transmitted by the flea is called Yersinia pestis by the species Genopsilla chiopis. And this Genopsilla chiopis actually carries Yersinia pestis, and Yersinia pestis causes the disease called plague, and the distribution is global. And different types of protozoa are also transmitted by different species of flea. And uh, in the completion of the life cycle of different cystode and nematode, flea has a role. For an examples, uh, different sort of fleas, for an example, dog flea, cat flea, or pulex irritants, human flea, or different rodent flea, they act as the intermediate host in the completion of life cycle of different cystodes, such as Diphylidium caninum, Hymenelophis diminuta, Hymenelophis nana. And in canine filariasis caused by Diphetalonema reconditum, also transmitted uh, by different species of flea. Just some side notes. So plague considered one of the most dangerous diseases in the world, and there are three major pandemic have been demonstrated. The first one is Justinian plague. This is named for a Roman emperor, Justinian. So Justinian plague, that is the plague originated in AD 541 in Africa and later spread throughout the Mediterranean region, killing an estimated 40 million people during 6th and 7th centuries. And the second pandemic is called the Black Death in 14th century, originated in Central Asia and spread to Europe as a consequence of developing trade routes between two continents. So in 1352, it killed around 25 million people in Europe alone. And later on, 1346 to 1353, killed 75 to 200 million people all over the world. And in 1665 to 1666, which is known as Great Plague of London, it killed one-third population of um, England. And the third pandemic occurred in 1896 to 1948. It kills more than 2 million people in India alone. So this is one of the latest reports showing the distribution of plague in different continents. So we should be more aware about the plague as well as its vector, that is Genopsilla chiopis. There are different means of transmission of Yersinia pestis from rat to human, but here I would like to discuss the transmission of Yersinia pestis from rat to human by the biting of flea. So Yersinia pestis is a gram-negative coccobacilli, is the causal agent of the disease plague. If you look at this picture, you can see the natural cycle of plague. Here, the Yersinia pestis is in the body of the different wildlife population, particularly the rat population, and this flea will feed on this uh, wildlife, and the cycle will be maintained here. This is called sylvatic plague. But in an outbreak situation, the wildlife population or rodent population may die, and this flea will seek for alternative source of uh, feeding host. There is alternative source of host, so they can move to feed on the human population or other domestic pets, and these domestic pets also bring this flea to the home. So whenever an infected flea feed on the feed on a host, whether it is rodent population or human population, whatever, they will suck the blood and the blood will go through the midgut. So if you look at this picture, you can see this is the esophagus and then proventiculus, this is the midgut. So after taking the contaminated blood, that is blood with Yersinia pestis, they will reach to the gut and the bacilli will start multiplication within the midgut. And in the second picture, you can see due to the continuous proliferation of the bacilli, this will start blocking the proventiculus and the lower part of the esophagus. And the third and fourth picture, you can see there is a complete blockage of the uh, proventiculus, as well as there is a rod-like extension of the plaque in the esophagus. So as a result, when these flea feed on a further host, they are unable to suck the blood in. As a result, they will become very hungry, they will become very maniac to feed. As a result, as they are unable to get the blood in, they start changing the host. And consequently, these maniac flea start depositing organism to different individuals. As a result, they are actually transmitting the Yersinia pestis to rat to human population, or one individual to another individual. So diagnosis of flea infestation is very easy. If you see flea dirt on the body coat of the animal, you can easily assume that this animal is suffering from flea infestation. Or if you can see the lesion associated with flea allergy dermatitis, you can assume the flea infestation. But you should go for the differential diagnosis with the mite infestation. For doing so, you have to take the skin scraping, and after processing, uh, you should examine the screen scraping under the microscope, and if you see a specific mite uh, based on their morphology, you can consider whether it is due to the flea or the mite. And for the laboratory confirmation or species identification, you have to collect the flea, and after processing of the flea, if you observe them under the microscope, based on some characteristics, you can easily identify at least the genus level or species level of different flea. So in earlier part of my presentation, I have talked about the morphological features of the flea. So there are two types of Tanadia or comb. One is genal comb, another one is pronatal comb. So if we divide the flea in three groups, that is flea without comb or without Tanadia, so there will be Echidnophaga, Pulex, and Genopsila. Flea with pronatal comb only, so they are Ceratophilus and Nosopsilus fasciatus. And flea with genal and pronatal comb, so there could be Tenocephalus canis, Tenocephalus fellis, and Sphenopsilus. But in our you can identify based on different characteristics. Sometimes it is very important to differentiate between these two, Pulex and Genopsila. So all these three uh, flea, there is no comb, that is there is no pronatal comb, there is no genal comb. So based on some other characteristics, you have to differentiate which one is which. So in case of the Pulex species, there is a bristle here. This is called ocular bristle. So if the ocular bristle is located just beneath the eye spot or eye, that would be considered as Pulex. Or in case of the Genopsila, if the ocular bristle is located in the front of eye, then you can consider it is Genopsila genus. And it is also important, as Tenocephalus canis and Tenocephalus fellis are very common, so you can identify based on their general combs. So if you can see, the first spine of the general comb is smaller than the second one, that flea would be Tenocephalus canis. And if the first and second spine is completely equal, that might be considered as Tenocephalus fellis. Control of flea infestation. Okay, so the control of flea infestation includes treatment of animal to kill the flea and elimination of different developmental stages from the environment and prevention of uh, prevention of the reinfection. So for the treatment of the animal, that is to kill the flea, different insecticides can be used. The first one is very popular one, flea collars. This is actually the plastic strip impregnated with different insecticide. And what sort of chemical can be used? They are, the popular one is dichlorobos, meladion, and sometimes combination of the dichlorobos with phenitropine uh, can be work better as a flea collar. Nowadays, there are a lot of commercially available flea collars that can be used for both flea, tick, and the prevention of the mite. And the duration varies. Some of the company claims their flea collars can give the protection up to 7 months or 8 months, but on an average, you can consider it the duration of protection may be 3 months or so. And insecticidal shampoo can be used for the killing of the flea. And another means of killing of the flea include dusting or washing with the insecticides. For an example, hexachlorocyclohexane can be used for dusting and washing in flea infestation of the dog, and Comofos can also be used. Tropical application is also a popular one to kill the flea. Ronal 1% solution can be used. And if there is heavy infestation, the dipping can be another alternative way of killing the flea. So Ronal 5% with 4.65% dichlorobos can be used. You could remember that dichlorobos is a acetylcholinesterase inhibitor. That is, they will paralyze the uh, insect, or they will paralyze the flea. So the next one is for the poultry. So what sort of insects are available in the farm or infesting the poultry? Depending on that situation, spray or dusting can be used. The popular one is hexachlorocyclohexane. It could be used for dusting as well as for spray directly on the floor, directly on the farm premises, or on the birds. And how often you will go for the spray or dusting, it depending on the intensity of infestation. So obviously not very frequent. Depending on the situation, it could be every 4 weeks interval, but do not spray 7 days before slaughter. And the second step for the control of flea infestation is the elimination of developmental stages in the host environment. So the premises should be thoroughly cleaned, that is removal of the hot spots. So hot spots are those places where the eggs, larva, pupa is more. So premises should be clean, should be thoroughly cleaned, and if there is any cracks or crevices, that should be vacuum cleaned to get rid of all the flea, larva, eggs, and pupa. And afterwards, after cleaning the cracks and crevices, an effective insecticide should be blown to prevent the breeding or to kill the rest of the leftover pupa or adult or eggs. And bedding of the pairs should be cleaned or destroyed. And the third effective way of controlling the flea infestation is the prevention of reinfection of the environment. So if you should go for the treatment of the animal, first one to kill the flea, and the second one elimination of developmental stages in the host environment, you have to do it. Third one, regular cleaning of the animals. hygienic measurements. If there is a chance of flea infestation, then you should start using some kind of dusting or powder or shampooing, etc. So these are the reference proof from which I took a lot of information to prepare this presentation, and I have also used different figures, images from internet. And thank you all for watching this video, and I would like to request you to stay with the channel to get more interesting videos on veterinary entomology. Thank you.